Welcome back to Biomindscape. Today we are going to know about an experiment by Joshua and Estin Linderberg in 1952, the replica plating experiment. So what was this experiment all about? The scientists wanted to know are resistance bacteria like antibiotic resistance they are formed before exposing to an antibiotic like spontaneous mutation or any are they formed because of the antibiotic forces which mute them like induced mutation. So this replica plating it helps this answering all these questions. So basic idea, if the same colonies they survive in the same position on a selective plate, they will form mutation happening before selection. So let's see the steps which explains this replica plating method. So first is to make a master plate that is non-selective medium or bacterial culture. It is spread on a normal agar plate without any antibiotic. Many colonies grow and this is called the master plate. All type of bacteria grow here, normal plus the mutant one. Then the second step, press a velvet cloth on the master plate. A round sterile velvet cloth is gently pressed onto the plate and the velvet picks up a few cells from every colony like a stamp or a Xerox copy. The velvet now has a pattern machining the master plate. That's the drawing of the cloth touching the plate. Then third step, press the same velvet on the second new plate. Replica, non-sensitive non-selective plate and replica selective plate. The replica non-selective plate where no antibiotics and all the bacteria they can grow. Whereas if we see the replica selective plate, it contains antibiotics or phage and only resistance bacteria will survive. So the bacterial transferred are in the same position as on the master plate. The fourth step is it is the incubate the plate. After incubation, on replica non-selective plate, all the colonies will grow normally and the pattern is same as a master plate because no selection will takes place. And on replica selective plate, only mutant resistance colony they grow and they appear. They are exactly the same position as on the master plate. So what does this mean? The resistance colony it appear in the same place on every selective plate and the resistance cell were already present before you expose them to the antibiotics. So these were not created by an antibiotics. The mutation is spontaneous and non-induced. So what are the important application? It provide non-adaptive spontaneous mutation. It is used to isolate mutation that is antibiotic resistant, auxotropes, etc. And you can keep the original pattern of the colonies while testing on different media. So replica plate shows that mutation occur before selection and the location of the colony remain the same and on the selective and non-selective plate providing the mutant were already present there. So the replica plating experiment, the purpose of this experiment is to test whether resistance mutants they are pre-existing in bacterial population which arise spontaneously or are induced by which are exposed to a selective agents. So what is the principle of this experiment? If mutation occur randomly before selection, then the same colony, that is the same position that grew on a non-selective master plate will correspond to colonies that grow on the selective plate after replication. So if mutation were induced by selection, the pattern would be random. So what happens here? What are the various methods? So there are various steps which are done for this experiment for obtaining a result. So the steps are like growth of the bacterial culture and plated on a non-selective agar, and this is a master plate. So many colonies are formed here. So then the press a sterile velvet or a nylon replica tool onto the master plate and the velvet picks up a small number of cells at each colony position and imprint. Then touch the velvet to 2 or more new plates. So a replica, the non-selective plate that is control, and a replica selective plate that contains antibiotic, phages or other selective agents. Then incubation is done. All the plate they are under appropriate condition. Then they are compared colony position on the master and replica plates, and on the replica non-selective plate you should see the colonies corresponding to all master colonies that verify the transfer work. and on the selective plate, only pre-existing resistance mutants will grow, and their locations are compared with the master plate. So what are the results that are obtained? If resistant colony appear at the same position on a selective plate as certain colonies on the master plate, and those resistant mutants were already present before exposure or spontaneous mutation. So if resistance colony appear randomly, the position not match, it will not match master colony or inconsistent across the replica, which could indicate induction, but replica plate was devised to support the spontaneous mutation. Consistent matching supports spontaneity. So this experiment, it support the fluctuation result that mutation arise randomly and not in response to the selection. So what are the important points here? Always induce a replica non-selective plate to confirm the successful transfer, and if use multiple replica selective plate to confirm the reproducibility. Sterile technique is essential, and contamination can give false positives. For low frequency mutants, you might need many master plate to detect them. So what are the applications here? Demonstrate non-adaptive, that is spontaneous mutation in microbe, widely used to isolate mutation, example antibiotic resistance, phage resistance, and auxotrophs, and useful in the microbial genetics, evolution, and biology and lab selection protocols. These are the some of the applications of this experiment. So what do we understand by this? The replica plate test whether the resistance bacteria it arises before or in response to the treatment. So we plate a culture or non-selective medium, that is a master plate, and then press the velvet to transfer the colony imprints to a selective plate, and after incubation only pre-existing resistance colony grow on a selective plate at the same position as on the master. So matching position should show mutation where was spontaneous, and a key result in microbial genetic which proves the mutation are random and not induced by selection. There are two models of how resistance might arises, like a direct and spontaneous. So direct or induced mutation model. In this model, it shows the mutation occur because the virus is added. The virus force bacteria to mute, and so resistance colony should appear in random places on each selective plate. Whereas spontaneous mutation model, it shows mutation occur randomly before virus expose, so a resistance colonies should appear in the same position on every selective plate. So here in this diagram we can understand the direct mutation in response to the virus. Here the master plate is all the colonies look the same and the virus is added later. If the virus induces resistance, then the resistance cell will appear randomly and the replica plate are made. Resistance colony appear in different spot on different plate and there is no fixed pattern. So this hypothesis is incorrect, whereas spontaneous mutation where before the virus has been added, the spontaneous mutation, here what happens, the master plate, here the some colonies arises, they have mutants like red dots and these mutants were present before the virus were added, and when replica plate are made using the velvet, the resistance colony appear in exact same position on every replica plate, so the pattern is identical. So here the hypothesis is correct and supposed by the Linderberg's experiment. It is not different from the earlier diagram, so this is the same replica plating experiment, but the diagram explains two possible explanations in simplified comparison format. If resistance colony appear in the same place on every selective plate, mutation happens before selection, which causes the spontaneous mutation. Thanks for watching. Do like, share and subscribe.